Good evening, everybody. My name is James Ferrier. I'm the product development manager at Nufarm Agriculture Canada. So my role involves looking after our research and development team, our technical support team, the team that runs our 1-800 line, as well as our research farm team. Dan Colcook, whose name and contact information should be at the bottom of the slide here, would be your sales rep for your area, focusing on the horticulture, turf, and IVM portion of our business. Dan can't be here today, so he asked me to step in and talk a little bit about our high and low bush blueberry segments. So, Nufarm, rapidly growing company. We've been in Canada for about 25 years here now, but we started out in Australia over 100 years ago as a farmer-owned fertilizer cooperative, slowly expanded into pesticides, and in the 50s or so, expanded into 2,4-D, other phenoxies, and kept going to, we're one of the larger manufacturers of glyphosate now in the world, and we've kept up with our older passion of working on fertilizers, working on biostimulants, and we've been continually building our capacity to manufacture older off-patent products as well as newer patented pest control products. So our horticultural lineup has grown rapidly over the last 5 to 10 years through our partnership with Valent and other manufacturers. So we have a whole suite of insecticide, herbicide, fungicide, and PGR products for most of your horticultural crops. I know today's focus will be blueberries, so talk mainly about Danitol, Chateau, and Wash. Starting off on Danitol. Danitol's a relatively newly registered product in Canada. That registration's been about 3 or 4 years now, but it was extremely needed by growers to help target spotted wing Drosophila and other insect pests in your cane fruit and low-growing berry crops. So it's fenpropathrin, so group 3 insecticide, a wide range of rates for use with that product. But the really interesting thing about it, again, is that spotted wing Drosophila control. So this is one of the only products where you can actually target your fruit that's fallen on the ground to get control of that recruitment of pests that are coming on that fruit that's fallen on the ground and started to rot. So you're allowed two applications per year with this product. And there are some maybe less than ideal REI and PHI information on it, just due to the fact that it's a fenpropathrin molecule there. You can use it on your tree fruit, your blueberries, both high and low bush, fruiting veggies. And I'll give you some of the information here on your re-entry interval. And again, a little bit less than ideal, but 17 days for handset irrigation, 15 days for tying or training your raspberries, and 13 days for hand harvesting or scouting in your low bush berries. 3 days for mechanical harvesting and 24 hours for other applications or activities in the field. Again, nice wide window of insect pests with Danitol. So everything from blueberry maggot to cherry fruitworm, your typical mites, your European red mite, spider mite, Japanese beetle, leafhopper, 2-spotted spider mites, and most common weevil pests. And again, the big reason we brought this product forward was through the minor use process. We were requested to bring this product in by many of the berry grower groups just for that spotted wing Drosophila control. So there's really nothing that's quite as efficacious as Danitol. So you can use that product basically as soon as you start seeing your first pest activity, and repeat it a 14-day interval. Don't exceed that 2.3 liters per hectare per season. So that would be 2 applications at the high rate. And again, you might look to direct that spray towards the soil to control insects that were falling on or present on fallen berries. So it's probably a little bit more relevant to the high bush guys or to guys growing cane crops and that kind of thing. But grain, again, great product for that spotted wing Drosophila problem. Talk a little bit about Chateau EZ. So this is our flagship herbicide for Nufarm for a lot of our crops. We have this registered in everything from cereals to soybeans, corn, berry crops, palm fruits, other tree fruits, veggies, and really unique group 14 herbicide. Very strong activity on small seeded broadleaf weeds and smaller seeded grasses as well. It's got really nice sort of long, strong residual control after application, which makes it really stand out as far as its length of residual and range of activity on just a wide range of different pest species. It's a liquid formulation, which makes it really nice to use. Anywhere from 46 to 182 mils per acre, depending on soil type and length of residual that you would like to get. And it's also very tank mixable. So you can put it with your glyphosate, your glufosinate, your carfentanil products, pretty much any other herbicides that you want to mix with. Chateau will mix well. We like to see it applied in the fall. A few reasons for that, but one is that if you apply it in the fall, you're going to get better activation of the product, and you get longer and stronger residual into the next year. So you can apply it in the spring for some crops, but we find that a lot of growers like to apply it in the fall for your berry crops. Interesting thing with that weed control is that Chateau has very low solubility in the soil. So once you apply it, it sticks in that top layer of soil and stays around for an extended amount of time. That's what sort of builds up our long residual activity. So see really good control on, again, all those sort of small seeded broadleaf weeds, your pigweeds and lamb's quarters, chickweeds, all that kind of stuff. Some moss control as well in there. But dandelions, kochia is obviously a western weed up there, but your fleabanes and things like that. We also get some control on wild buckwheat, wild mustards, foxtails, barnyard grass, different thistles kind of thing, cleavers. So again, just a nice range of broadleaf and grass control from a pre-residual standpoint. So again, the way that product works is it forms a layer on the soil, so that controls the weeds as they germinate. It stays there as long as you've had moisture for activation for the better part of the year. Part of my work on the development team, we looked at this product out to 16 to 20 weeks after it being applied and still seeing really nice residual control of a lot of key weeds. Some weeds, such as those in the pigweed species, you'll see control for even longer at some of the higher rates of the product. Interesting thing with it, though, is once you disturb that soil layer, you dilute that product and you lose control once you've disturbed that layer, but you can then plant essentially any crop the following year if needed. So again, very low solubility, so you see low potential for leaching, which is especially important for those of us in the eastern part of Canada when we worry about water flowing into the St. Lawrence or into the oceans and local creeks and streams and whatnot. So we do find again that when that product gets used in the fall, we see really nice activity. You get it down, you get it in contact with the soil, you get snow cover over the winter. That snow melts in the spring and activates that product. And we find that it's even better activation than 1 or 2 rainfalls in the spring. So, and then it's there first thing in the spring to start getting those early emerging spring annual weeds. Wide range of weeds again on that label, but specifically on the low bush label, 61 or 90 mils per acre. You're going to pick up your fleabanes or lamb's quarters, ragweed, dandelion, mosses, different nightshade species, pigweeds, and those grasses again, foxtails especially. Needs to be applied to dormant berries. So don't make more than 2 applications in a growing season, a minimum 30 days apart. So we'll see growers often put that product down in that sprout year and the dormant year, spring, and then fall. Also see it as a dormant post-harvest spray, the year of harvest. And growers just, like I say, they find that fall application seems to work really well. So again, that fall piece is something that we really harp on. Another reason for that is that we do see better crop safety with that fall application, especially in some of the more sensitive crops like garlic and asparagus. And again, it's a great way to get a head start on your weed control for the following season. So if you can get that product down in the fall and get extended residual into the spring, it can help spread out a little bit of your workload and avoid having to get out there first thing in the spring and get herbicides down. So again, that residual can last well into the year. So controlling those weeds in the fall also has other benefits besides just a clean crop. So we know that those weeds can steal nutrients and moisture that those crops are going to need in the following spring to get going. Winter annual and perennial weeds and biennial weeds that come up are much easier to control in the fall. And if you can take them out in the fall, you don't have to worry about them in the spring again. So that's a key point. We've done a lot of research work on our research farm and through contract research organizations, looking at rates of products needed to control weeds in the fall. And generally, especially with perennial and biennial weeds, if you can get them in the fall, lower rates of herbicides are needed to control those weeds and get much better results than if you'd let them continue on their growth and tried to target them in the spring. So another benefit of that fall application is that you can really target and tailor your herbicide program to the field, to the specific pests that you have, and make sure that you're doing your best to manage your field and spread your equipment time and your employee time around. So the other thing, maybe not quite as big of a deal for the berries, but we do see it in field crops as well. We see a lot of weeds that play host to different insects and diseases, and that carries over. So if you can control those weeds in the fall, then you have essentially eliminated those hosts for those insects and diseases. On to fungicides for the Nufarm portfolio. Again, for both high bush and low bush berries, Quash is our product of choice. So metconazole, I'd say this is our product for mummyberry and quite a few other berry diseases. Again, low and high bush product, broad spectrum, reliable, ultra low use rate. So we're using anywhere between 73 to 118 mils per acre. Nice package size there. I'll do 18 to 30 acres per case. And again, it tank mix as well. We have a pretty good formulations team at our lab down in Morrisville, so North Carolina, sorry. And they are always working to help optimize formulations and make sure that they're going to tank mix the way we need them to. So if we need to put her down with a little bit of liquid fertilizer or another pest control product, we're confident in the ability to do that. So nice thing about Quash is that it is locally systemic, and you do see some translaminar movement as well, and you get some curative and contact activity, which a lot of other products will not give you. So on our label, obviously the big one being mummyberry and anthracnose ripe rot. You also see early blight, Fomphosis canker, suppression of white mold, cherry leaf spot, brown rot, blossom blight, and fruit brown rot, suppression of powdery mildew, and suppression of scab. So the way quash works with that curative and systemic activity and preventative activity, you can apply it anytime from penetration all the way to onset of symptoms, and you're going to get activity. You're going to get control of your main diseases. Whereas with your strobilurins, you really need to use them before before that disease is penetrated into the crop. So nice thing with quash is being able to get that both curative and preventative activity with it. So we've done a lot of monolinea trials with quash over the years, and it's always just a step ahead of products like Tilt. And the interesting thing about metconazole is it does provide some plant health benefits as well. So you see essentially water use efficiency increases, and just the plant's going to grow a little bit more vigorously and better able to fight off stresses. See that particularly with potatoes with quash. Changes the potato crop's physiological growth, so it actually puts more energy into the tuber, less into the upper growth. So you see shorter, darker statured potato plants with a larger number of tubers underneath. The same principle applies to other crops it's used on. It just increases that plant's ability to reproduce. So, again, the mode of action of quash, it's locally systemic. So you see some translaminar movement. You see some movement outwards within the leaf, although it's not phloem mobile, so you don't see it moving up and down in the whole plant. Again, I talked about that stress preventative application. We see metconazole providing changes in how a plant grows to help it better react to stresses. You see that movement in the foliage and that locally systemic and translaminar movement. Again, quash doesn't move in the phloem, so it doesn't move up and down. It just moves sort of outwards on the leaves. You don't see it moving from roots to tops or tops to roots, but you do get movement within treated leaves. So if your spray coverage isn't great and you still hit that leaf at some point, it's going to move around within that leaf. So on blueberries, recommended rates typically 76 mils per acre for your anthracnose, your mummyberry, and your Fomphosis. Apply it, when disease development is sort of starting. Typical timings for your specific disease, and you can make reapplications at 7-day intervals if needed. I recommend not to make more than 3 applications per season. So through our R&D program and discussions with some of our sort of extension people and whatnot, we came up with a little bit of a recommendation here for low bush program, including quash. So recommend applying quash at late bloom for that mummyberry control. Second app around 7 days later for mummyberry, botrytis, anthracnose, and alternaria with pristine. And then a third app with quash around 7 days after that, again, for that mummyberry, botrytis, anthracnose, and alternaria. So with that, I'd like to thank you for your time today. Again, glad to be here on behalf of Nufarm. Dan Colcuck, I think he was speaking to you guys earlier today already, but he is your rep for the area. He's glad to take any questions via email or phone if you need to get a hold of him for anything. He's always glad to get out in the field, as am I. So if you have any questions for me now, I'm glad to take them. Otherwise, thanks for your time.
Yeah, thank you, James. That was perfect. Does anyone have any questions for James?
There was one there, Chloe. I'm just trying to find it.
Yeah, there is one in the chat. It was kind of a back and forth. I think someone kind of also chimed in. So one person just said, Danitol is a broad spectrum pyrethroid. How do we balance its benefits in pest control with the risks to beneficial insects and pollinators?
Well, following your best management practices are always important. So, we definitely don't want to be applying during bloom is the big thing, and that is on that product label. So make sure that we are approaching, our economically viable pest level before we even think about spraying. And obviously, with all these products, are, I'm making my living selling pest control products, but we don't want to use them needlessly. We need to make sure that we're following an integrated pest management approach to get in there. So.
And typically with spotted wing and maggot, there shouldn't be much in the way of blossoms in the field anyhow. So.
No.
One request I'll put into you is that maybe you can ask your team to approach both the Oxford Frozen Foods and the Wyman's group to see if you can get your product put on the user list.
For which one? For
Both.
Danitol?
Oh yes, sorry, for Dantov. Yeah, because we do have a very small list of products that we can use for SWD.
All right.
And since the spotted wing season seemed to start before the crop did this past year, having more tools in the toolbox is a positive thing.
All right. So Oxford Frozen Foods and who else?
And Wyman's.
Wyman's. All right.
Because both of them have what they have is a restricted use list, I guess you call it.
Yep.
And if it's not on that list, then it doesn't matter whether it is or isn't registered.
All right. We can
can't
certainly
be
approach
used.
them because we've done all the work for residues and all that kind of stuff. So there shouldn't be any concerns there.
Yeah, and that's all I think they need is to make sure that it's usable in all other countries that they would ship products to.
Yep.
And now would be the good time to sit down with them because that list goes out usually sometime in March.
Okay.
So
I can I manage our MRL list for all our countries that we see product get exported to, so I can make sure that that gets looked after. So.
Perfect.
that'd be excellent. Yes, thank you.
And someone also commented Millens would be useful as well to
Oh yes,
see,
melons too.
see if it was appropriate for their markets as well.
Great. Good evening, everybody. Thanks very much to Terry in particular for the presentation. Terry asked me to give a talk on pollinators and bees, and he thought the one that I gave at the Wild Blueberry Producers Association meeting, I guess it was 2 or 3 months ago, would be useful. Some of you may have already seen this presentation. I apologize for any of the repetition, but there's always value in seeing things more than once. So hopefully we can learn something new, even if you've seen it before. So this talk is all about bees, and we know that pollination is really important for lowbush blueberry, of course. And decades ago, Gordon Kinsman pointed out that inadequate pollination is probably the most important cause of poor yields in blueberries in Nova Scotia. And that's probably not always the case. Of course, there are a lot of issues ranging from weather to diseases to weeds to insect pests that can affect yields. But I think we all agree that pollination, if you don't have pollination, you're not going to get very good yields. And if you don't have insects pollinating your crop, you're not going to have any fruit. In wild blueberry, the pollination system is unique and well, not completely unique, but fairly unique and complex. And I think it's really important to understand that system to appreciate the pollination challenges and pollination expectations for your crop. I'm going to spend a little bit of time here talking about that system. And it's important to understand that wind won't work. So for a lot of crops like wheat and corn, etc., even deciduous and coniferous trees, a lot of the pollen is blown around with wind. That's not sufficient for wild blueberries. The reasons for this include the fact that they have poricidal anthers. And the anther is the male part of the flower that releases the pollen. And they're poricidal in the sense that they're kind of like a salt shaker. And you need something like an insect to get in there and agitate that anther to get the pollen out. You can see here that the flowers hang downwards. They're not even really exposed upwards to the wind. And the pollen is quite heavy. They have these pollen tetrads that do not disperse well in the wind. So an insect is really key to get that pollen moving around. We need to remember that insects are not moving that pollen around for you exclusively. They're not even moving it around for the flowers. They're doing so for themselves. They use that pollen as a protein source, and they use the nectar as a sugar source. And they're doing so rather haphazardly. So what that means is that they can move, for instance, pollen within a single flower. So if you see right here where I'm pointing with my mouse, this is a situation where a bee might have that pollen on its body, and it's moving that pollen from the anther to the stigma, which is kind of the female part within the same flower. In another case, you may be like over here, you may have an individual plant with multiple flowers, and the bee is moving the pollen from one flower to another. But that's still considered a self-pollination situation. The flowers are on an individual plant, and the pollen is being moved within that individual plant. On the other hand, you can have pollen moving between flowers on different plants, and this is what we consider to be cross-pollination, as you can see right here. So the insect is moving the pollen around. It's not really deciding if it's moving within a flower or to different flowers. And that has ramifications in terms of blueberry clones, because it is the clone that is the unique, genetically unique plant in your field. It's not the individual stems, but that clone. And you can see this picture right here of a typical blueberry field with lots of clones. And these can range in size from small to 5 meters squared up to 25 meters squared. Now, what's important to understand is that a lot of these clones are self-incompatible. In fact, over half of them in most fields are self-incompatible. What that means is if you get pollen moved from one flower in a clone to another flower in a clone, it's probably not going to result in a viable fruit set. So you need to move pollen between clones. Interclone pollination is usually what's required. And that's significant when you think about how bees move. So this figure right here shows that, a clone could be about 200 centimeters, so a couple meters across in diameter. But bees fly flower to flower at a much shorter distance. And you can see here that most bees are flying, only 20 or 30 centimeters between flowers. And what that means is that a bee is often moving around within a clone. And when you have self-incompatible clones, that might compromise fruit set. Now, not all clones are self-incompatible. There are some that are self-compatible. And Frank Drummond did some really nice work down in Maine a few years ago. And all I want you to see right here is that on the bottom, we have clones that bloom early, clones that bloom in a little more in the middle of the season, and clones that bloom late. And you can see that there are a fair number of clones in the early bloomers that are self-compatible. So some clones are self-compatible, other ones are not. Pollination is complicated by short flower receptivity. When that bloom opens, you've only got a short amount of time for that flower to be pollinated. After about 4 days, the success of pollination really drops. So it's important to get the bees on those flowers that are blooming early. And you also have to contend with the fact that you've got sometimes different species in your fields. There are actually about 4 or 5 different species of wild blueberry, and 2 are prominent, right? We have Vaccinium angustifolium and Vaccinium myrtleoides, and these 2 species are not compatible, but they often exist side by side. And you can show that this can be a problem by doing a simple experiment as we did right here. You can take pollen from the same clone and do inbreeding, You can take pollen from 2 different clones of the same species. So go from Angustifolium, for instance, to another Angustifolium clone over here. Or the bee might be moving pollen from Angustifolium to Myrtleoides, and that has significant effects on things like fruit set and berry yield. If you outcross between 2 different clones of Angustifolium, represented by the A right here, or 2 Myrtleoides, you're good. You get good fruit set and you get good berry weight. But once you cross between these 2 different species, you get low fruit set and low berry weight. And if you self them within the same clone, it's even worse in terms of fruit set and berry weight. So all this is to say that the pollination biology of low bush or wild blueberries is complicated. And fruit set is usually pretty mediocre. It's not unusual that it is, because most plants produce a lot more flowers than they do fruit. But if you look at wild blueberries, and this is some work that a former PhD student of mine, Andonia Milofopoulos, that some of you may know, did. And here we're looking at the numbers of flowers during bloom, the number or the amount of fruit set for those flowers, and the amount of berries at harvest. And what you should notice is that across both years, you get a continual drop with only, around 40% fruit set. This has been shown by some colleagues of mine in New Brunswick. Here we're looking at fruit set on the y-axis, and you can see it's, around 40, 50, 55%. And we get the same thing, but some work done by Frank Drummond down in Maine. Here we're looking at proportion fruit set, around 40, 50% or so. So, the take-home message is that, if you can get 40 or 50%, that's quite reasonable and quite good, actually. And if you get anything higher than that, around 60% or so, that would be excellent. So the pollination system in wild blueberries is complicated. So when we talk about pollination and pollinators, we always think about bees, and we should, but I just want to remind people that there are non-bee pollinators. And several years ago, I was inspired by this paper right here, looking at pollination in cloudberry. Back in Newfoundland, we would call it bakeapple. And this is a crop or a plant that occurs in a lot of the same systems as wild blueberry does. And I noticed that they looked at nocturnal pollination, and they found that that can result in significant fruit set, which made me ask, well, can we get nocturnal pollination in wild blueberry as well? And indeed, we can. I did a small experiment to show this, just setting up cages, as you can see right here. And I simply allowed some of them to be open all the time. Some of them were closed 24 hours a day. Some of them were only open during the day, and some of them were only open during the night. And what we find is that with the cages that were only open at night, we still get significant fruit set. Not as much as the cages that were open all day long or open 24 hours, but still significant, resulting in good ripe berries and good berry weight. And when we look at the insects that are moving around at night, there are a fair number that are carrying blueberry pollen. You can see right here, these are small beetles, weevils, that you may notice when you do sweep netting in your fields. Around a third of them were carrying blueberry pollen. You get very, very common moths like geometrid and noctuid moths. A significant number of those are carrying pollen and so on, including different flies and parasitoids. So bees are certainly the most important pollinators, but there are other things that are out there in your field that are crawling around in your flowers, and they are moving blueberry pollen around. But the bees are most important, so let's talk about them. First off, let's consider wild bees. And there are a lot of wild bees. I've done a number of studies on this in wild blueberry, as have others. And this study right here back in 10 years ago now, we found about over 90 species of wild bees in wild blueberry fields in Nova Scotia. So that's almost half of all the bees that we know of in Nova Scotia. There are probably 200 species of bees in Nova Scotia. Again, about 90 of them we found in blueberries. And this is only a partial table right here. And a lot of them are around your fields, and they are there during bloom in some cases. For instance, we have this Andrena corallina, quite prominent during blueberry bloom, which is highlighted by the gray bar here. We have this nomata species of bees that's quite prominent during bloom as well. And a lot of the other ones, the numbers tend to go up in terms of trap captures a little bit after bloom. But the take-home message is that blueberry habitats in Nova Scotia really harbor a rich community of wild bees. And they're not evenly distributed in fields. Fields vary greatly, of course, in terms of the habitat around them, in terms of the size. And what we generally find with wild bees, and here we're looking at the abundance of bees on the y-axis across different distances. And you can see that, when you go into the woods, there are hardly any bees there at all. But as soon as you get to the edge of your field, you find lots of wild bees. And as you gradually move into the central portions of your field, the number declines. It still stays pretty high, upwards of about 100 meters. You're still getting a fair number of native bees out in fields. But once your fields start to get bigger than that, we start to see a decline. And you get changes in the diversity of bees at different distances. And bees are kind of like airplanes, is the analogy I like to use, right? When you fly long distances, you need a bigger aircraft. And bees are the same thing. And you can see right here that as the distance from the field increases, you only tend to find larger bees further out into the field. So the smaller guys tend to hang out closer to the field edge, and the larger bees are further out into the field. We also know that wild bees are very good pollinators. And Steve Javerick, who is a research biologist down at Agriculture and Agri-Food Canada in Kentfield, did a really nice, very highly cited study all over the world on wild pollinators. And I'll just show you some of the results from his study. And, things like looking at the numbers of flowers visited per minute. For instance, whereas, something like a honeybee right here will move to about 8 flowers per minute, bumblebees will move to 11 or 12 flowers per minute. So they're actually moving faster around the blossoms. When you look at the amount or the number of pollen grains that are deposited in a single visit, you can see that whereas something like a honeybee deposits around 11-ish or so pollen grains per visit, bumblebees and even things like the Andrena bees, these are ground nesting bees, will deposit from anywhere from 30 to 50 pollen grains per minute. So they're moving around the flowers faster and they're depositing more pollen. And when they do visit a flower, the percent success of that visit is quite high as well. Bumblebees have a very high success rate. When they visit a flower, almost 100% of the time, it's getting pollinated, not so much with things like honeybees. About a quarter of the time, it results in pollination success. Now, this doesn't mean, of course, that honeybees are no good. Quite the opposite. They're very good at pollinating blueberries, but it just means on a per individual bee, bumblebees are more efficient than most other bees that are out there. And we'll talk about honeybees and bumblebees a little bit later as well. So wild bees are important, and that means that we should be doing as much as we can to conserve and boost those populations. And lots of people have looked at this in many different cropping systems, and we've looked at it in blueberries. Rob McCollum, a former PhD student of mine that some of you may know, has done things like tried different types of planting, different clovers and facilla and whatnot, and showed how they can attract bees, as well as things like natural enemies, which can be important in pest control around your fields. Robin did lots of trap nesting studies, so creating these kind of bee hotels, if you like, to see how they might attract natural bees. And indeed, when you put these out around your field, here's the number of cap nesting tubes. That just means that they're occupied It's kind of like closing the door to an occupied hotel room. You can see that the numbers of cappings goes up during blueberry bloom, so they will occupy these nesting sites. And we need to remember as well that there are lots of bees that are actively nesting directly in your fields. And I love this picture from one of Frank Drummond's former students. This is a blueberry field that was burned. And you can see all these little patches right here. These are soil nesting sites for Andrena bees. You might recall that this is a genus of bees that's very good at pollinating blueberries. And you can see this field is just completely peppered with these nesting sites of this bee. So habitat is key, and it can exist around fields or directly within fields. And people have done work to see if, we can create habitats to conserve those bees. And these can be called pollination reservoirs. This same student of Frank Drummond's in Maine looked at things like clover, natural regeneration, and wildflowers. And basically, all of these bees like these options, but they have different preferences. So you can see things like bumblebees and honeybees prefer clover, whereas things like the solitary nesting bees seem to prefer more of the wildflower mixtures. And things down here like these syrphid flies, and yes, there are flies, they're called hover flies that can pollinate blueberry. They tend to prefer things like the natural regeneration. So there are lots of different options to try and conserve bees around fields. In any case, results can be mixed, but in this study, they found that when you have these pollination reservoirs, you definitely get more bees on blueberry blossoms, and this can translate. It can take a little bit of time, a few years to build up those populations, but you can get better fruit set in the fields that have those pollination reservoirs. Okay, so that's the wild bees, but of course we understand that there is a need for managed bees as well. And again, decades ago, Kinsman wisely pointed out that although wild bees are around your fields and in your fields and that they're important, their populations are pretty susceptible to fluctuations from year to year, whether it's due to weather, whether it's due to disease and parasites. And because of that, you should hedge your bets by considering introducing things like honeybees or bumblebees into your fields. And I don't think there's any doubt that honeybees can be of tremendous benefit to your crop. There have been several studies looking at this. This is one out of Quebec. And they actually placed 500 hives at the end of a huge area of blueberries that extended 6.7 kilometers. And right here, they're just showing that the honeybees go pretty far, right? So, I mean, you see significant numbers of honeybees up to about 3 kilometers away from those hives. And then only then do you start to get a decline in the number of bees. So they can go quite far into blueberry fields. Native bees are also there, of course, right? And it's a steady number of native bees right across the field. And they can complement each other. But what's clear is that as you have the honeybees and more honeybees, this can translate into better fruit set. So you get in this figure right here, we get a significant relationship between the numbers of honeybees and fruit set, and a relationship between the numbers of honeybees and berry weight, as you can see on this bottom figure right here. Up to a point, of course, right? And what they showed here is that at around 15 or so honeybees per 2 meters squared, so going around in the field and counting the bees on the blossoms and then coming back later and collecting the fruit and weighing them. After about 15 bees per 2 meters squared, you start to see diminishing returns. So bees definitely can be important, but up to a point. Honeybees, that is. Another study closer to home, this is with Leonard Eaton and John Murray back in 1997, where they looked at a bunch of fields in the Maritimes and in Maine. And again, showed that there's definitely a benefit of having honeybees. These asterisks next to these numbers just mean that there's a significant improvement, if you like, right? So with honeybees right here in this column, a significant increase in the amount of fruit set, a significant increase in the percent of berries, and they noticed a real correlation between fields that had honeybees, and the more honeybees tended to have better yields compared to other fields. But they did note that there's a lot of variation from field to field and from year to year. And in fact, when you look at a kind of a little bit of a follow-up study that Leonard Eaton and one of my other colleagues, Villas Nams, who's also retired now, did, where they looked at around 500 fields of various sizes, from 1 hectare up to almost 40 hectares over 5 years. They essentially looked at the numbers of honeybees and the yields of those fields, and found that there was a relationship, but not a super strong relationship. You can see here, all of these dots represent different data points. So we're looking at the numbers of hives and the yield. And there is a positive relationship, but only up to about 4 hives per hectare, okay, or about 1.6 hives per acre. After that, there's just a lot of uncertainty, a lot of variation that makes the relationship not very strong. So honeybees can work, but only up to a point. I mean, you can see right here in some fields where you have 4 hives per hectare, you get very, very good yields, and other ones where you have 4 hives per hectare, the yields are terrible. So just, again, just a lot of variability, even though honeybees certainly do work. Bumblebees are also available commercially now. For the past few decades, we've been able to purchase bumblebees from companies like Copert and BioBest, and they also work. You'll recall that, looking at wild bumblebees, they're very efficient as pollinators. And people have done studies looking at how bumblebees compare to honeybees, and wild commercial bumblebees compare to honeybees as pollinators. Frank Drummond, again in Maine, has done a couple studies in this area. And in this one, they looked at fields that had about 3 hives per acre, and they had different stocking densities of bumblebees, looking at around 1.25 to 2.5 quads per acre. So this is pretty well what, Copert would recommend. They recommend about, 1 to 2 quads per acre. And indeed, we see that they're pretty comparable. So here we're looking at the number of bumblebee colonies against, 3 or 4 honeybee hives per acre, and there's not too much different. There's variability, of course, from year to year and endpoint to endpoint, but all of these A's right here above the bars mean that there's no statistically significant difference in the amount of fruit set, the weight of the berries, etc. So honeybees and bumblebees can do a great job. Another study, again, looking at the same honeybee density, 3 or 4 hives per acre versus 1 bumblebee quad per acre. And again, quite comparable in terms of the amount of fruit set, the amount of mature berries, and the berry weight, which was significantly greater than when you only have wild bees. So the addition of those commercial pollinators definitely made a difference. I'll finish off here talking about how many bees do you need, and this is a bit of a perennial question, right? I mean, you have to decide as a grower, should I get honeybees? Should I get bumblebees? And if so, how many should I get? This comes with obviously experience and understanding your field, appreciating the strength of the honeybee hives that you might be getting from your beekeeper, the weather, the amount of bloom that you can anticipate in any given year. But several rules of thumb have been suggested, and I'll just quickly remind you of them here, but these are available if you look on fact sheets for Nova Scotia, New Brunswick, Maine. You can find this information. I'm happy to share it as well. Back in the day, Kinsman recommended, if you have at least 1 bee per square meter with decent temperatures, and that's probably pretty good. It's still probably pretty good, but, since Kinsman's time back then in the late 50s and 60s, of course, production and expectations for production in wild blueberry have gone up a lot. So this probably doesn't hold very much as a rule of thumb anymore. Another kind of rule of thumb is to go out into your field and set up a few different quadrats and do counts in your fields on a warm, calm, sunny day. And over a period of 5 minutes, if you don't see any wild bees, then obviously you've got insufficient pollination at a pretty high level, and you might need as many as 5 hives per acre. If you, over a 5-minute period, if you count a couple bees per meter square, then maybe, you've got a pretty good natural pollination force, and 1 hive per acre might be sufficient. A final rule of thumb that I'll mention is one that was developed by Frank Drummond several years ago. This one's a little more complicated, but not that complicated. And this is a predictor of the percent berries. So if you count blossoms, you can predict the percent berries you're going to get based on the numbers of bees. So this is the number of honeybees that you count in a square meter per minute times this number, 7.8, and the number of native bees that you count per minute. And this should give you an estimate of the percent berries that you can expect later on in the season. Of course, a challenge with these rules of thumb is that there are at least a couple. I don't know if they've been rigorously tested, and that's something I would like to do. And the other thing is that they depend on you going out and counting bees in your field during bloom. And that's a little bit late in the game, right? I mean, by that time, you might go and count the bees in your field and say, oh, I don't have enough bees, I need honeybees. But it's too late to contact a beekeeper and get bees into your field. So what I think is needed is a better predictor model for the natural pollination force to give you an idea of what you've got out there to gauge the need for commercial bees. So in conclusion, I think I'm almost out of time here anyway. Remember, wild bees are really good pollinators. They're significant contributors, and that there are things we can do to bolster their communities. And always think about conserving their populations. But undoubtedly, honeybees and bumblebees can significantly increase fruit set and yield. And most growers, if you've got larger fields especially, should definitely consider that investment. Remember that there's a lot of variation across fields, clonal species composition, the size of the field, the surrounding habitat that can support the natural pollinator community, the year, depending on how cold the weather is, how much snow cover we get, how wet the spring is. All those things can affect the natural pollination force, which can affect the need for commercial pollinators. And we do have models that are out there to predict that natural pollination force, but as I already discussed, they probably have some limitations. That's all I have. And I'd like to thank Terry, especially for the opportunity to present to you here today. And over the years, I've had lots of people, I'm sure people on this call who I've collaborated with and people who have helped support the research that I do. And I'd be happy to answer any questions if there are any.
Thanks, Chris. That was an excellent presentation. Good reminder for us.
Yeah, very interesting. There is one question in the chat that I'll read to you. So with climate change affecting bloom times and pollinator activity, do you think blueberry growers need to rethink their pollination strategies?
I don't know if they need to rethink it a lot in the short term, but they need to be thinking about it, right? So this goes back to my ideas or my point about predicting the pollination force. I think with climate change and just the unpredictability of our climate, we don't yet know how many wild bees you're going to have around your field. I think we all agree that people will go out and say, oh, I see lots of bumblebees this year. And the next year, oh, I don't see very many bumblebees. The question as to why that is as it relates to things like the weather is unclear. And what I think would be useful is to develop a predictor model that takes into account for an individual field the floral habitat, but also the climate that season, and to use that as a proxy to predict the natural pollination force. So, climate change is definitely going to affect bees. I don't think in the short term, though, growers need to change anything major, but just be very mindful of the fact that it can change the ball game, so to speak.
Perfect. We've got one more here that just came in. Is it more beneficial to put all of our pollinators on the field at once, or is it better to stagger the hives employed?
I think staggering has merit. It's obviously more complicated logistically. If you're talking about honeybees, they will make these concentric circles, and then they will move out around fields. And the more familiar they become with their habitat, the further away they will go. And that means they will have decreased fidelity for blueberries or blueberry bloom. And in that sense, if you can introduce, a new clutch, if you like, of honeybee hives to a field that are not as familiar with the field and surrounding landscape, they're more apt to kind of stay at home. Now, there are challenges with that, but if you are able to stagger things a couple of times, I could see that as having value.
Chris, I got one quick question for you.
Yeah.
So many of us have what you call an edge effect quite often, where you don't seem to get very good pollination along the outside edges of the field.
Yeah.
But having listened to your discussion here on native pollinators, and they seem to be within 200 meters of the woods edge, why do we have poor pollination then along the woods?
I never thought of that. I mean, I wonder if it has something to do with, I was going to say things like shade and changes in microhabitat around the edges.
Yes, I can see that, right? Like, I mean, for the bumblebees and the honeybees, I think they like it warm or
Yeah.
stay out in the open. But I'm just thinking if the native ones are adapted to that 200 meters around, then you would think that they should be less bothered by shade and cold and stuff. So I'm just curious why then we do see that edge effect so often.
I'm not sure, although that's really interesting. Maybe I'll talk to you later about that, Terry.
Sure. No, it popped in my head, so I thought I better
Well,
pop it out before I
I'm
lost it.
thinking about it in terms of a research question. So that's yeah, I never thought of that.
Well, thank you very much, Chris. We appreciate your time for this evening.
Okay.
Yeah, thank you.
Thanks, everybody.
You seeing presentation mode?
Yep, looks
It's
great.
good.
Looks good. Okay. I can't see my notes, so we'll just run with it. All right.
Sounds good.
Is any consolation,
Okay.
we can't see your notes either.
Okay, well, that's a good thing. That's a good thing. All right, here we go. So full disclosure, I thought about pouring some blueberry wine tonight, but I didn't. I'm having a rooibos tea, being a good girl right now. So my name is Alana. I'm a horticulture innovation manager with Corteva. And this is my agenda. These are the products I want to discuss tonight. A couple herbicides with you guys, and some insecticides, and finish off with some questions. So the first herbicide will be curb. And so this product, selective pre-emergent or early post-emergent product, and used to control annual and perennial grasses. Fescues is one of the types of weeds that it's strong on. Liquid suspension, and the packaging is in 2 by 10 liter jugs. So the mode of action for this herbicide, it's inhibiting root and shoot development in germinating seeds and young seedlings. Symptoms, and I didn't find a good picture of this, and so my apologies, but the symptoms are the roots will turn club shaped or swollen, and this product is absorbed by the roots. It is soil active. There is a little bit of, well, not a little bit, there's a lot of a xylem systemic activity, which means it's going to move upwards into the plant. But you're not going to get very much leaf absorption activity. So the foliar application, that application of leaf absorption is limited. So these pictures are just showing, again, the importance of making sure we're getting great soil contact. And the purpose of using curb is to prevent the development of the root system. So some key application guidelines. Apply it in late fall, October to November. So again, the timing is not set in stone. It's dependent on temperature, ground freezing. We need our soil temperature to be colder. We need it 10 degrees. We want it above freezing, but cold. And we want to avoid temperature swings. So again, a fall application, but making sure we've got proper conditions. You know, a little bit of moisture, so applications followed by rain or even a bit of wet snow are good. Some other application guidelines. This product, you'll see great results if it's applied to sandy or medium textured soils. Muck soils, you guys really don't have to worry about that too much in Nova Scotia for wild blueberry production anyway, but there is reduced efficacy on the muck soils and high organic matter. So just something to keep in the back of your mind. Optimizing performance. So with curb, make sure that all the fruit is harvested. Application is permitted crop year or sprout year. There is a rate range as per flat fan not.
We seem to be losing you there, Alana.
I'm back. Can you hear me?
Yeah, we can now.
Yep, you're good now.
Okay, what did you last hear?
But a minute back.
Okay, so I didn't lose you too much. That was really
No.
weird. Okay, but that's the fun of
Yes.
virtual. Okay, so can I share? Let's try this again. Okay, are we back?
Yes.
Perfect.
Yep. Okay. So water volume, again, I was just saying water volume is important with curb. There's no rain fast period because this is soil active. And just keep in mind one application in a year. Next herbicide will be Launtrell. And active ingredient, I say clopyralid. That's how I remember this one. Everybody's got their own pronunciation for this one. Group 4. And your packaging 4 by 2.67 liters. Control of vetch, clover, and various clovers, but vetch is the important one here. Spot treatment. So Launtrell, keep in mind, we're doing spot treatments. You can either use a backpack or boom sprayer, but you know, we're doing spot treatments with this. And Launtrell, it's the sprout year only. And since most of the time we're targeting vetch with this, I mean, just make sure we're aiming for early bloom. June is generally the time period, but you know, just keep a watch on your fields. Rain fast 4 hours, you know, re-entry 12 hours, PHI 12 months. So 10 months, my apologies, 10 months. So again, with this product, there is a rain fast period, just something to keep in mind out in the East Coast. Third herbicide I want to talk about is Steadfast. Now registered on Blueberries. So again, this is new as of 2024. I believe you guys have had one full season with Steadfast. The components of Steadfast, it's 2 group 2 herbicides. So this is rimsulfuron and nicosulfuron. So these herbicides, active ingredients, you should be familiar with. as they are what was in Altum. And unfortunately, Corteva did discontinue Altum, but Steadfast is your replacement. And just to review, the strategy Corteva had for Steadfast was to first get low bush blueberries on the label. That was key. We wanted to make sure we had a replacement product for Altum. And now the next step is to increase the weed listing. And so, I mean, Steadfast, the history of it's a corn herbicide. And so the weeds, green foxtail, wild oats, volunteer canola, volunteer wheat. Not really what you're going to find in your fields, but just know that we are working on increasing the weed spectrum. I can tell you that actually Scott White and our regulatory manager are communicating quite frequently with ideas for new trials and ways for us to get the label expanded. So stay tuned on that. So keys with Steadfast, again, application guidelines. There's a rate range, generally use the high rate. Non-ionic surfactant is needed, so make sure you're adding something like Agril 90. Water volume, key here again, minimum is 15 gallons of water per acre. You do have that PHI, you do have rainfast that you got to consider. We're going after our annual grasses, and ideally we're aiming to make sure those grasses at the 1 to 6 leaf stage, and 1 application a year, and it's in your sprout year. And so make sure, if we can get it early, prior to a lot of blueberry or heavy blueberry emergence, that helps with increased crop safety. Prism, next herbicide. We're just breezing through this. So the next herbicide here, Prism. Active ingredient, rimsulfuron. So group 2, again, it's one of the components that we just discussed in Steadfast. Your packaging is 12 by 480 grams. Your weeds, Prism's been around a little bit longer. We got a wider weed spectrum on Prism, but still your grasses, right? So barnyard grass, witchgrass, quackgrass. So we've got a wider spectrum on this product. Your rate, 60 grams to the hectare. A surfactant is also needed again, so make sure you've got some Agril 90 in the shed. Minimum water volume, 15 gallons to the acre. So similar as Steadfast, but Prism, as always, double check that label before you're using. And weed, we want our grasses, same 1 to 6 leaf stage. And we're applying the spring of the sprout year. That's what I had for herbicides. And so what I'll do is I'll jump into our insecticides now and just keep flowing, and hopefully we got no more technological issues. So Entrust insecticide, this has been a standalone product for Corteva. It is part of our spinosins group. And so our spinosins are Success, Entrust, GF120, which you guys may not use too much, but used a lot in the apple industry, and Delegate. And so our spinosin group, we've got 2 active ingredients within this class. We have spinosad, which is Success, Entrust, and GF120, and we have spinetoram, which is Delegate. So Entrust, what you're getting with this is our certified organic product. And I know there's a few low risk and certified organic blueberry growers. If you are not one of those, Success is another alternative, but with the organic growers, it is Entrust. Listed by EcoCert Canada. The way this product is working is contact and ingestion. So you've got 2 forms of the way this product is going to work, which is great because the way I look at it, I know we're all doing really great application. We've used our little sticky cards, and we know we've got great spray droplet application. So we know contact is going to work, but not all the time are the insects on the top of the leaf, right? So sometimes they're on the bottom. And by having ingestion, we know that when they start munching, we're going to still get control. So that's what I like about Entrust. We got a 1-2 punch here. We're getting 12 by 1 liter bottles in the package. So again, with Entrust, with the spinosyn group, it is a naturalite product. So what that means is it's its own class. This is a group 5, which makes it fantastic for IPM, rotating your chemistries, all things that horticulture growers always think about. And you know what this product contains, it's derived from a bacteria. So this is a fun game on your Friday night. I want you guys to remember to say, how many times can you say this active bacteria ingredient? Saccharopolyspora spinosa. Now, after you've had a few, that's the game. Saccharopolyspora spinosa. This active ingredient is made by a fermented process. So again, it's a bacteria, it's a naturalite product. And the way it's working is it's exciting the nervous system of the insects, and it's leading to death, which is what we all want on those insects in our fields. So one of the best things about this product is it is safe on beneficials. You know, your big-eyed bugs, your damsel flies, your ladybugs. So if you guys, we just had a great discussion about the wild pollinators. Entrust is great for making sure we've got a robust IPM program. Now, that being said, when this product is sprayed, when it's wet, it is toxic to the pollinators. So we're not spraying at bloom. But once the residues are dry, then we've got a non-toxic product to the honeybees. Things Entrust is registered on. So spotted wing Drosophila, spanworm, flea beetle, and blueberry maggot. So, you know, all these key pests that you guys are dealing with in your crop, you can consider using Entrust in your rotation. Make sure you're following the rate range. Again, high rate range or high end of the range if we've got high insect pressure. But really, you're following your IPM program. You're starting the application of Entrust when monitoring tells you to, and we're starting small. So things like spanworm, Entrust works better when the insect is small, as opposed to, you know, a larger, like 4th instar larva. Application guidelines with Entrust. Again, just follow your re-entry interval, 12 hours. You got rain fastness after 2 hours. Minimum retreatment interval, 5 days. You're allowed 3 applications. So, you know, work it into that crop, the pest rotation, work it into your spray rotation. And generally we say don't make more than 2 consecutive applications of group 5. And tank mix compatibility, we've got the generalized tank mix statement on our labels, so you are good to go with most things. The final product I want to talk about is Delegate. So again, as discussed before, it is in our spinosyn group. Fruits and vegetables, the label, we all love to read labels. You know, when you've got some time, read the Delegate label. It is a robust label. We've got lots of insects listed on Delegate and all sorts of crops. Long-lasting control, and I just want to highlight that. Spinatorium, the active ingredient of Delegate, has a longer residuality than spinosad, because Spinatorium is more photostable. So we will be able to get a longer residual. I tend to say Delegate will give you 7 to 14 day residuality, whereas Entrust with spinosad will give you 7 to 10 days. And again, there's all sorts of things that go into figuring out how long a product will stick around. But that's the general rule of thumb, is you do have longer lasting control with Delegate. Safe to beneficials. So again, it's a naturalite, it's part of our naturalite group. So same as before, making sure that we're not applying during bloom, and after bloom, we're safe on the pollinators. When this product is dry, it is safe and non-toxic to honeybees. What we're controlling, spotted wing, flea beetle, and spanworm. Again, rate range, high rate. So again, use your IPM practices, follow along with, as if the population is high, we go high rate. And timing, obviously we want to get in early, but only spray if your monitoring is showing that the insect pest is there. Similar reentry intervals. Again, Delegate and Entrust are very similar products. But what I tend to say again, so you got your longer residuality with Delegate, and it's with the way that the spinatorium works, it will work a little better on the larger insects. So if you don't get in until third or fourth instar of the insects, so the insects are bigger, go in with Delegate. It can handle the larger insects. What else? Excellent tank mixability as well. For more information, jump onto our website, corteva.ca. Our horticulture section is vast, lots of little tidbits of information on there. We also have a field guide app. If you don't have that, scan that QR code, grab the app, and it's stronger on the row crop side, unfortunately, but there is horticulture information there. And I'll finish with questions. Are there any questions?
That was great. Thank you very much.
Yes,
You're
thank you.
welcome.
So there is one question that came in. Is it better to apply curb pre or post fall pruning?
Pre or post fall pruning? So well, no, I mean, I am not as familiar with low bush blueberries. So
Normally it
if
would be
you're
done post mowing.
post mow, because I was going to ask, how late are you guys going with the mower? Like, I mean, are you
There's
going
some that
into
might push
November?
the limit, but you'd normally try to get the mowing done well ahead of the application.
Okay, so yeah, like I was gonna say make sure it's going post, but I mean, if you guys are pushing the mower into December, well then, that's your call. But yes, thank you, Terry, for that help.
No problem.
There is one more here. So one person was just wondering, Elena, if you could put the QR code back up.
Oh, yep. Okay, so let's share. Okay. Share, and can I go backwards? There we go.
Perfect. And then one other person just said, if these products get wet from, say, a little rain, can they be harmful to bees?
If these products get wet. Oh, no, once they're taken into the plant, no. No.
All right.
Yeah.
Perfect. And
The app is on the website, so there is a QR code. So if you feel free to again jump on the website, and yeah. So
perfect.
that's perfect. All right. Well, I am gonna say goodbye to you guys, and enjoy the rest of your meeting.
Yes.
Thank you very much.
Thanks,
All right,
Elena.
take care.
Oh, it's nice to be involved in the presentation. So thank you very much for the invitation. And what I thought I'd better do is bring in some help tonight, just given we're in the middle of the semester. So I'll have 3 other people that I'll be helping with this presentation tonight, consisting of Naomi Jackal, and Naomi's doing her master's with us. And then I have 2 postdoctoral research fellows in the form of Kenneth Anku and Oladameji Pupulo, who will also be making presentations. So the research itself would not be possible without the support we received over the years from the producer associations in Nova Scotia, New Brunswick, and Prince Edward Island, as well as the Bragg Group. And also what I'd like to mention is the help of Mellon Farms is also appreciated, because there have been times when to get a data package ready for a new pest control product, other crop groups are required, and so they've helped out in the acquisition of strawberry data when required. So that's very much appreciated. Along with that, the public sector has also been very active in supporting the research program over the years, as well as some of the agrochemical companies. And the ones listed there are the ones that have directly contributed to research activities over the past year. So what I'd like to do tonight is give an overview of what our priorities are for the research program, just give you a bit of insight on where things are pointed in the upcoming year. I'll provide a little bit of information on some of the pest control products and situations with MRLs that we're a little bit concerned about, some challenges as well as opportunities in this upcoming year. And then after that, Naomi, Kenneth, and Ola will be proceeding with their presentation components. So as far as the research program is concerned, it very much is one that, especially after we ran into this problems with the leaf diseases, so blueberry rust, followed by septoria and also spherulina and valdensinia, the research program really started to screen products on a fairly active basis starting approximately 20 years ago. And we really haven't stopped since that period of time. There just seems to be ongoing challenges that come up that necessitate this. And along with that, we have research underway that's really trying to focus on looking at the production system, because one of the beauties of it is it's such a low input system compared to other fruit production systems. You know, we're using native stands of blueberries that have been there for a long period of time. No tillage practices are used. Also, supplementary irrigation is very rarely used, at least in the Maritimes. And so we really want to have a look at the footprint associated with producing a kilo of blueberries compared to other fruit crops, and that being wild blueberries. And I think what it'll clearly show, and we started this work back in around 2005 or so, is that it has quite an advantage when you look at it compared to other fruit production systems. Along with that, given the situation with climate change and all the challenges posed with it, we also want to have a look at, okay, well, what is the carbon sequestration potential of a blueberry field? And we know it's most likely going to be better than some of the agronomic crops, but not probably anywhere near as good as what the forestry sector sees. So this is something that we're trying to look at in more detail. Also, assessing the blueberry fields remotely with the use of unmanned aerial vehicles or the UAVs or drones is still an ongoing activity that we'll be continuing to proceed with. And you'll hear some updates on this. And then we're also doing some additional sustainability work, looking at life cycle analysis, biodiversity assessments, and then also some regenerative agriculture work. So to start things off, I thought it'd be good just to go back and look at this concept of the MRL. And so when pest control products are used in berry production, you will have trace levels of these active ingredients as well as their metabolites in treated products. And these are what's referred to as residues. And so what a maximum residue level is it's the highest level of a pesticide residue that is legally tolerated in or on food or feed when pesticides are applied correctly. So in other words, in accordance with good agricultural practices, and more specifically, the label involved. And so the amount of residues found in food that we consume, it must be safe for consumers and also be as low as possible. And so as part of the assessment process, in looking at the registration of a new pest control product, Health Canada, they must determine whether or not the consumption of a maximum amount of residues that are expected to remain in a food or food product when a pesticide is used, according to the labeled directions, will not be a concern, especially with human health. And the maximum amount of residues expected when it was legally established as a maximum residue limit, this is regulated by the Pest Control Products Act. And along with this, extensive collaborations occur with other international agencies, okay, and those being especially in the United States, and then also in the European Union, as well as other areas. So in Canada, as we're aware, it's possible for farmers with a valid pesticide applicator's license, okay, to purchase and apply these restricted pest control products for use in wild blueberries. And it's really important to note that a lot of work's gone into the development of these labels so that the approved use patterns should provide harvested wild blueberries are compliant with the MRLs that are established for that pest control product when this product is produced and for sale and for consumption in Canada. However, this does not necessarily mean that it's going to be suitable for sale and consumption in export markets. And presently, there's over 40 of these different export countries that are presently buying wild blueberries. And this is something that always is important to be aware of when we're going through the discussion of discussing pest control products. And this is a table that was actually put together by the North American Blueberry Council, and they update it on an annual basis. And these are some of the active ingredients that are on their list of concern is a good way to sum it up. And with the sale, some of the export markets of concern with harvested fruit include Korea, Philippines, Vietnam. And then when you go down through some of the triazoles, such as difenoconazole and metconazole, these are a concern in China as well as Vietnam. And then we start to get into things that are a little bit more directly applicable to the wild blueberry industry, such as novalurin, which is one of the actives in cormorant, propiconazole, which has been problematic for quite a few years in the European Union, fenpropythrin, I think is how you pronounce the active, if that's the active in danitol. And this has been a concern in the European Union. And the active ingredient, this relates to a previous question, it's not actually a registered active ingredient for use in the European Union. So it's one of the reasons why it's been a bit of a concern for use in the wild blueberry sector. And similarly with melifine, it's a similar scenario there as well. So with that, this gives you some insight then into why some of these active ingredients can pose problems to the wild blueberry sector. Along with that, and following up on some of the previous discussion with MRLs, what you'll notice in this particular table is when you compare their MRLs for the active ingredients, they do change as you move from Canada to the United States and then to the European Union. And a good example of this is with bosquid, which we don't see as much use with it. It's one of the active ingredients in pristine. You can have almost a complete full difference in the allowable MRLs. And then it's even more pronounced when you look at prothioconazole, which is the active ingredient in Proline as well as in Sortel, where European Union levels are minimal, so almost default with respect to the allowable levels there. So as a result of this, definitely need to be careful with the use of Proline and Sortel once you get up to that point of bloom and thereafter in the crop year of production. So it's always a bit of a concern as far as that's concerned. As I mentioned last November at the WEPAN's annual meeting, when you go through, and in particular on the fungicide side, you look at, okay, well, what is the present status of some of the active ingredients that are in use in the European Union, just given its importance to the wild blueberry sector? What you'll notice is actually the majority of the active ingredients, they're actually set to expire this year. And along with that, when you look at the active ingredient in Miravis Prime, trade name's adepidin, the active ingredient, I've always mispronounced this, is pitimeflumetofen, I think. It's still pending, and it's been pending for a few years, but upon talking to Cheryl Truman with Syngenta earlier today, she's indicated, well, there's indications that this MRL will eventually get established in the European Union. And that's fantastic news because it's been the thing that's been holding back on the use of this product. It's a really, really good fungicide. So it's good to see that move along. And then on the other end of the spectrum, with fludeoxonil, its expiry is in June, and my understanding is a vote is being held on the continued use of this active ingredient in the European Union. So it is a bit of a concern. May get approved, but may not. It's just one of the things that we'll just have to see how it goes. And so if you actually go on the European Union's website, you can just do a search on the active ingredients and get this information. So with prothioconazole, you can see here, it's set to expire August 15th. And the pitimeflumetofen, Syngenta's fungicide, it's still pending, as I mentioned earlier. And it's a great fungicide. So if we do eventually run into some problems with fludeoxonil, which is one of the actives in switch, this is going to be an excellent botrytis fungicide to look at once that established MRL comes along. So then when you look at a potential fungicide portfolio for use in the wild blueberry sector this upcoming year, not too many changes compared to previous years. The big challenge will be to see what happens with the status of some of the active ingredient registrations in Europe, and if they'll be renewed, or are some of these actives going to be phased out. So it's something that we're definitely going to have to keep an eye on. And along with this, when you look at the wild blueberry streams and the production practices that are used, we have our conventional stream. On the other end of the spectrum, there are organic blueberries being produced. And then we also have what's referred to as a residue-free berry, where if you know the characteristics of the active ingredient, along with that of the blueberry, the berries, you know when the last application window should be with these pest control products. So this is something that there's some movement on, and my guess is it'll something that will continue to expand this in the future, this production of residue-free berries. And so with that, when you look at the research program, we do have an active screening program, and it's also a program that is also driven by industry. And so really we go through 3 steps in the process. So starting off, as soon as an agrochemical company has a product of interest that they're interested in collecting some data on, might have an initial look at it. And then as it moves along in the registration process, it'll be moving into the phase 2, where we'll have a look at it and its use patterns and wild blueberry production in a little bit more detail. And then the 3rd phase is when we actually say, okay, we want to make sure this product is thoroughly vetted, just so that when the rubber does hit the road with its use in the wild blueberry sector, there's going to be no mix-ups. So it'll be used as intended. And so with that, a lot of these trials are actually coordinated by Bragg Lumber Company, and because we're using their fields, they're also providing equipment and management support as well, so field management activities with these trials. And this particularly becomes true when you look at as we're ramping up the assessment of these products, both with what we call strip plot trials, which is situated here, and then on a larger trial basis with the self-propelled sprayers. And it's very important because what you tend to see with some of these trials is you can run into some issues, as we have here with some phytotoxicity, and that's something that we want to try to catch earlier in the process than later. So when you then look at where we're presently at with respect to the pest control products that are registered or being used in the wild blueberry sector. There is most likely going to be changes that are going to be forthcoming, and a lot of this is associated with what's going on in the European Union. And as I mentioned, quite a few of the active ingredients there are presently under review and may not be re-registered. Okay, so as a result of this, when you look at our screening activities, a portion of them go through Bragg Lumber Company and their research and development program. And with that, it really started 20 years ago with some of the leaf spot diseases. So the Septorius, Ferrier, Valdensinia, and blueberry rust carried on into monolinea blight and botrytis, stem blights such as Phomopsis and Gadronia. We've also had some screening trials there. And then also, of all things, mosses. Trying to control them is what resulted in Chateau initially coming along. So with that, one potential product came up when I was at the minor use meetings last March just outside Ottawa. And what we had there was one of the companies, Bear Crop Science, approached us about looking at a new active ingredient called ecofolin methyl. And I didn't even know the name of the active until about 2 weeks ago. And what we were told then, when had the initial discussions, is look, it's novel, it's non-selective herbicide. And there was indications then that it was even somewhat of a new mode of action. Subsequently, it's perhaps not as novel as what we thought, given that it inhibits tubulin polymerization. But the beauty of it is a product that's going through the global registration process. So it's one of Bear Crop Science new active ingredients. It's been screened throughout the world, and it has a high level of efficacy against cold and warm season monocots as well as dicot weeds. Okay, so both grasses as well as broadleaf weeds. And the product itself was just recently patented in 2022. And when you look at its mode of action, it's actually most likely going to remain classified as a group 3. And when you look at it, some of the other herbicides that are in that particular sector include pendimethalin, which is Prowl, and I believe that's still, at least it's used in the US high bush sector, and propazamide. I was trying to think, okay, where have I seen that before? Well, it's actually the active in curb. And these products inhibit the polymerization of free tubulin dimers into microtubules, and subsequently they inhibit plant cell division. And as a result of this, it interferes with cell division, okay, so during mitosis processes, and also cell wall formation. So it's a really interesting herbicide, and I was warned in advance it's going to be quite different when you look at its effect on some of the weed pressures that we have. And it's a product when applied, it gets hydrolyzed into carboxylic acid icofolin, and as mentioned earlier, it's really been proven to be quite effective. And I'll show you some of the weed pressures that's had some efficacy on. And the unique thing about this, compared to other herbicides in that mode of action, is they have post-emergent activity as well. And that's one of the interesting things with this herbicide that we noticed is for the initial week and a half, you really don't see too much in the plots. But from that point onward, things really start to move along and advance quite quickly. Right. And from that perspective, it's quite an interesting herbicide. And so just to give you some insight, and this was up at the Upper Brookfield site where we had the trial out, you can see when we first set up the trial, and a few weeks after that initial application, the weed pressures on the top, where they're uncontrolled, were becoming quite established. And you just saw as we progressed into July and then lastly November, very, very much so pronounced. And on the bottom, where you can see some of the plots that had the Ecopholin active ingredient, just excellent weed control, right? And so this carried through, and I went up and had a look first week of December as well. And the treatments were holding, which is fantastic. And one other thing that I'll mention, because it's nowhere else in the notes, is Bayer Crop Science. They're probably going to move this along, if it does work its way through the Canadian system, as a co-formulation. And it'll be co-formulated with their herbicide, Allium. And I'm trying to remember the active in the Allium, but it's escaping me right now. But anyway, give you some insight into what's been found in some of the other trials. Here's some work that was done by Half and others. I think this is a project, if I remember correctly, it was done in Europe. And what you'll notice is they have both the active ingredient, the ecopholin methyl, and then the ecopholin. So they looked at both combinations applied to the trials. And overall, they found excellent weed control. And I'll show you the species that were involved. The only two that appeared a little bit problematic were common ragweed, and then cobbler's pegs, or also known as blackjack, which I don't think we have in our blueberry fields. So really, really good control. And the standard that was used in this case, or the check treatment, was actually curb. Okay, so compared to curb, which is in the gray bars there, which were quite variable responses, the ecopholin methyl really did a nice job. So nice broad array of activity, and this is out of the study that I believe was conducted in Europe. And those that are situated in black are the ones that efficacy wasn't that great on. And then as far as the results that we found in the trial so far, and this is just 1 year of data, 2 sites, and not a really large trial size. I believe they were 4 by 4 meters. What we found is excellent control of fescues, which is somewhat reassuring. And bunchberry is the only thing that really seemed to escape it. Goldenrod, it only suppresses somewhat. It's the goldenrod will I think come back, and then some of the other rushes and sedges also, it only suppressed somewhat. So it's something to keep an eye on. So it's not a miracle, but at least the nice thing with this particular new herbicide is at least it appears to have nice control of the fescue grasses. So as far as its use pattern, again, it's just an experimental herbicide, but it's really the first good news with respect to fescue control we've had quite a while. No visible signs of phytotoxicity. We paid careful attention, and this is what Bayer Crop Science wanted us to closely monitor, was just, okay, what's the effect on the blueberry plant? And we didn't find any deleterious effects there, whether it was looking at the plant health with use of the UAV system, or even looking at the dry weights of the blueberry plant. There's no deleterious effects. And as far as the status, the trial is going to be remain in the ground this upcoming year, so we'll see how it looks going into harvest. We've worked closely with the pesticide minor use coordinators in Nova Scotia, New Brunswick, and Prince Edward Island on making sure they were aware of this new active ingredient. And Denny Augustine, he's really helped facilitate and coordinate the discussions. He's even checked on how the PMRA would receive if we move this forward as a priority, and there are some positive indications illustrated there. So it is a chemistry that's undergoing global registration. Its initial focus on the Canadian sector, and especially on the hort crops, is on tree fruit and vine crops. And this was done just from a amongst other things, a risk management perspective. And then they gave us the opportunity to have a look at it from a minor, or from a screening perspective. And so if we can move it forward as a priority, we might be able to expedite this process. However, just please be mindful, we have to have it work its way through the minor use priority setting workshop. So hopefully it'll go forward there as a priority. And it is going to take time. So I think, if I remember correctly from the literature, the earliest this product will be registered is 2028. So it's a little bit of a way yet in order to see this through, but hopefully it'll something that will come along. So with that, what I'd like to do is pass it along to Naomi Jackal then to give an overview of her research activities.
Okay, hello everybody. I'm just going to request control from you.
Okay, so I'll just go to my screen sharing. Let's try to see. Okay, can you I'll approve the sharing and then you should be all set there, Naomi.
Okay. So, all right. Just give me one moment. I can share my screen. All right. Can everybody see the presentation?
Yep, it looks good.
Okay, fantastic. Hi, I'm Naomi Jekyll. As David introduced me, I am a current master's student at Dalhousie University, and my research revolves around biological fungicides and specifically targeting Botrytis cinerea in wildflower bush blueberries. I did a trial this past year, so I'm a year into my research, and I'm here to share some of my results with you. And hopefully we can all learn a little bit together. So when we're talking about biological fungicides, they really play a key role in resistance prevention and residue control. They're strong in resistance prevention when it comes to diseases because they have really complex modes of action, including the use of competition, as well as the secretion of anti-pathogen secondary metabolites. Those two modes of action are harder to overcome and negate by the pathogens, which help them with the resistance prevention. Additionally, since the active ingredients for these products tend to be biodegradable and they are naturally occurring, they can be oils, they can be bacteria, they can be fungi, depends on what their target is. They allow us to have no residuals left after application, which helps with that residue-free berry, which, as David pointed out, is really the end goal when it comes to exports. The biggest disadvantage that we see when we're using biological fungicides is that when there is heavy disease pressure, they tend to not hold up, especially when they're used alone. As David mentioned, the European Union is a leader when it comes to making decisions on how the fungicides on the active market are being used, just because they are such a big importer of products. Looking at the fungicides specifically used against Botrytis cinerea, some on the chopping block for this year potentially could be fludeoxonil. Fluopyrim is also in review, as well as pyraclostrobin. And then next year, pyrimethanol and cyprodinol is also going to be reviewed. Not that we're overly worried for some of these, but some of them, as David said, specifically fludeoxonil this year, we'll have to wait and see what the final what the jury says about our uses of that active ingredient. At the end of the day, the European Union, really their goal is, and for most of Any Health Canada, they want a residue-free berry to ensure health benefits. But here, as we are working in agriculture, we want products that are able to be effective at the same time. So my work this last year was a field trial with the objective to determine the efficacy of tank mixing conventional fungicides with a biological product, Serenade Opti. I had 2 field sites, one at Fox Point and one at Diligent River. The target was Botrytis blight. So we had 2 foliar sprays, May 24th and the 3rd of June. So they were about 10 days apart. And then we had multiple stem assessments after, looking at the plant growth, the staging, as well as the disease incidence and severity and how that progressed. So we'll be looking directly at the data and what I was able to recover from that and make some conclusions about it. The treatment list that I was working with specifically, I tried to cover a broad swath of active ingredients. So I looked at an untreated control, Luna Tranquility, Sensation, Propulse Switch, Maravon, and Moravis Prime, and I tested them with and without Serenade. A lot of the time during our crop year, Propulse and Maravon tend to be staples, especially because their active ingredients are still approved. As David has says, Moravis Prime has the pidefumatofen active ingredient, which makes it a little dicey to use in the crop year since the EU currently is pending on its status as far as an MRL goes. But I wanted to include it because it is an excellent chemistry, which you will see. So there's 2 collection points for each site, which we'll go over. So this is the first collection point of June 11th. It was 7 days after the second application of chemistries. The untreated control and the Serenade Opti both had high incidence and severity of Botrytis cinerea. The untreated control, I was happy to see that because that means I got data collection. And the Serenade Opti was also high, and that has to do with the fact that, like I said before, biologicals tend to not hold up under heavy disease pressure, and this is evidence of that. The treatment that stood out the most at this point was the Moravis Prime, with or without Serenade Opti. They were pretty much equal. And then all the other treatments lean towards being rather similar. And even when you were looking at treatments with and without Serenade Opti, they pretty much fell in the same ballpark at this time around. Our second data collection point was 14 days after the last application. Untreated and Serenade both had high incidence and severity, which was good to see. Moravis Prime was outstanding in this case, quite low. But the chemistries that I wanted to point out that I thought were doing rather well despite it all were Maravon, and then surprisingly for us, we were happy to see that Luna Sensation and Serenade Opti, that mixed together, looked like it was doing well. That being said, overall, looking at the management of the disease, looking at the chemistries on their own and then mixed with Serenade, in most cases, there was a lot of the time that the tank mixes actually performed slightly less well when you're looking at the raw values, although statistically they were all overlapping and roughly the same. This is our second site, so this is Diligent River. This is back to 7 days after the last application. It was also had high levels of incidence and severity of Botrytis cinerea in the untreated and Serenade, which was good to see. Once again, Moravis Prime was at the front, lowest levels of Botrytis. Maravon was really close on its heels, whether or not it included the Serenade Opti. And Luna Sensation, once again, on its own, was doing decently well as well. Moving on to 14 days after the second application, you can see that there was a drastic drop in the untreated and Serenade Opti levels when it came to Botrytis cinerea infection. That has to do with the fact that this field had reached the FH stage, so when the corolla falls off. All of our assessments are done visually, so we take stems out of the field and we look and assess percentage of how much and how severe the Botrytis cinerea infection is. So we're looking for the visible signs of it. Because of the corolla drop, this reduces visibility a lot. So when looking at this data, it gives us a lot of interesting points, but I wouldn't take it as it is and only it. So comparing different fields over different years is really important. But looking at this data, Maravon still performed really well along with the Moravis Prime. Propulse, when mixed with the Serenade Opti, had a surprising drop in incidence and severity. And once again, the Luna Sensation was doing well as well. So after saying all of that and noting that a lot of the time that tank mixes didn't work, you're probably sitting there and are wondering, what are you supposed to take away from this? So I'm going to pull on past success from our research lab. This is some of Joel Abbey's work. He was a PhD student here before my time. And he did some work on the rotational application to include biological fungicides. He was looking at them in rotation with switch. So he did treatments of just the biological fungicides and then treatments of biological switch biological, and he tested a bunch of different variations. And he noted that the biologicals on their own had a 50% reduction in Botrytis cinerea presence compared to the control. But when you did the rotational applications, it reduced it by 78%. So just having the biological included with a single conventional fungicide and then another biological caused a significant decrease in the Botrytis cinerea levels. And he was able to conclude that when you use this rotation, you're able to match the effectiveness of 3 conventional fungicides used in a row. So in summary, for my specific research for the tank mix experiment, there do seem to be a couple minor points of interest. I'm going to continue to look into it as I have another year of study before I finish my master's. So points of interest are luminescence and propulse. Although at this point, the tank mix isn't showing any large benefits. The treatment efficacies, obviously, Marevis Prime ended up being a front runner, but as David has said, since pideflumethofen is one of the active ingredients and isn't currently registered, it's still pending in the EU, its use is a little dicey, especially when it comes to the crop year. Otherwise, Maravon did really a stellar job. It did, and Propulse and Luminescence all had their good points. Pulling from past years, the incorporation of biological fungicides into a rotation to help with IPM really shows only benefits. Not only does it increase the mode of actions that you are applying on a field and help with resistance management, there's evidence and it's been shown that it doesn't decrease the efficacy of your rotation at all. And finally, it really reduces the residuals that you see on the field at the end of the day. So looking forward, my research, I'm doing more repeated field experiments regarding the field trial that I did and I showed you the data for, and we're hoping to do a few other trials to help tease out some of the reasons why, and hopefully next year we'll be able to get you some more data on and some reasonings why this is working, or maybe we'll find that some other things are working better. And then I'm also looking into some botrytis blight sensitivity tests when it comes to some of these active ingredients. So we're hoping to determine if there's any real resistant populations here in Nova Scotia. So I'm looking forward to getting you that data. And so, yeah. Moving on, I think Kenneth will take over. But thank you.
Yeah, thank you, Naomi. Can you hear me?
Yes, we can hear you.
Okay. Yeah, hello everyone, and good evening. Once again, my name is Kenneth, and specifically I'll actually be talking about greenhouse gas reductions in wild blueberry. And there's a trial that we actually conducted last year in some of our research trials. So I'll just move straight to the point, even as we delve into this discussion. So we know greenhouse gas has actually been a topical discussion for some time, and we know its effects on climate change. And interestingly, agriculture actually places contributes significantly. And here in Canada, it's considered as the largest source when it comes to the greenhouse gas emissions. And when we talk about agriculture, we're actually talking about crop production, we are talking about animal production. And specifically, there might be other aspects that we can talk about, but for this presentation, I'm just trying to narrow it down onto these specifics as the main contributors, and data actually specifies that animal production actually contributes about 51% to the 10% that I'll say collectively they are contributing to the pool. And crop production actually contributes 28%, and then on-farm machinery, that is 21%. Someone would say these percentages are quite low. However, when you look at the total bulk of it, it's still quite significant. And in trying to see some of the practices that leads up to these levels of emission, we are talking about the application of fertilizers, inorganic fertilizers, farm machinery, we're talking about burning, decomposition, and many other activities that we actually carry out on the field. And these largely contribute to these emissions on the field. And we'll be wondering, okay, so what's the way forward? Can we actually stop using these fertilizers? Actually, no, we might not be able to stop using these fertilizers, but there are other means by which we can actually adopt to either reduce the amounts of fertilizers that we apply onto the onto our fields, and the other techniques as well, which I'll try and speak about this evening. And in trying to achieve this, there are some techniques that have been explored, that have been discussed. And tonight I would actually be focusing on one specific one, that is the use of microbes. And these are, we have plant and then soil microbes that are actually either added to fertilizers and applied in the field, and they actually help improve nutrient uptake and then also help in its efficiency on the field. And so I would actually try talking about the microbes for this presentation. However, moving forward into today's presentation, the next speaker will try and talk about something we popularly call as the prescription maps, which is actually fed into tractors or maybe drone sprayers, and they help probably reduce the amounts of fertilizers and all of those things. So those are some techniques as well, which will be discussed shortly in this presentation. There is also the aspect of humates, and these are not fertilizers, but these are organic materials that enhance nutrient uptake by the plants. Because of their properties, they are more or less, I'll say, soluble. So sometimes you can actually add them to these fertilizers and then apply to the field. And basically, as I said, they are not fertilizers, but they are enhancers. They help enhance nutrient uptake by the plant. There are also other techniques that are actually being looked into, and so we can talk about some amendments and phosphates as well. Recently, I was just looking into other ones, and so you could actually talk about nanofertilizers that are also on the scene, that people are trying to research into and see how best they can be effective on some of our fields. Now, I've mentioned these microbes, and for this presentation, I would actually be looking at three main ones, as I use those three in my research trial. And so I would first of all talk about the Gluconacerbacter, the Azotrophicus, and it's actually a very long name, but I'll simplify it and then call it as the nitrogen fixing bacteria, because it fixes nitrogen. So over here, you could see that this organism is actually applied. And then what it does is it helps to trap the volatilized nitrogen that is in the atmosphere. It helps traps it into the leaves, such that the plants can actually make use of it. So that is one of the microbes that we would actually be talking about in this presentation. Secondly, we will talk about the urease inhibitors and what happens when we actually apply our fertilizers. They go through a hydrolysis process where the urease enzyme actually breaks them down. And through this process, we realize that volatilization occurs, and then we end up losing ammonia into the atmosphere. We are losing our nitrogen. And then there are further processes that takes place where we have the nitrosomonas all occurring on the field. And what they do is they actually reduce these forms and make them actually escape, making them non-available to the plants. And so in using these microbes actually help prevent some of these processes, or they actually slow them down, enabling the plant to actually make use of this nutrient. And through research has shown that when nutrients are actually applied to the field, when fertilizers are applied to the field, the wild blueberry plant actually makes use of about 44 to about 67%, meaning that there is about 33 to about 53%, which is not actually used. And so adding these microbes, we could help improve their uptake by the plant, such that at least we could reduce the levels of emission. And so we realized that some research has been done, and this research actually shows that the incorporation of some of these microbes helps reduce the cumulative loss of nitrogen in the soil. So it still buttresses the point that these microbes have some role to play in the aspect of reducing the amount of nitrogen that we use, reducing the volatilization of some of these nutrients that we lose from the soils. And so in trying to conduct this trial, I performed two main trials. That is one, using the nitrogen fixing bacteria that tries to draw nitrogen from the atmosphere into the plant, which commercially, I would say a product being called the Envita or the Envita product. I used this product. And so this trial was actually conducted in two trial locations. We have Mercidon and then Mount Tom, where these trials were set up. And for my fertilizer products, I use the liquid fertilizer that is Alpine for this particular trial. And so the biological organism, as I mentioned, Envita, was actually used. And to set a trial, I used these treatments, a control where no application was actually made. And then I used different rates of the Alpine fertilizer. So I have a 20 kilogram of nitrogen per hectare, 40, 60, and then 100. And what I did with the setup was I actually applied the Envita, that is the microbe, to half of the plots of the setup. So more or less, it was a split plot design where there would be some kind of an interactional effect. And on the second trial, I tried to, I would say, assess some greenhouse gases, as I made mention from the start. These actually contribute largely to our climate change. And so I set up a trial again in Kemptown and then in Adamsfield. And in this trial, I also used the liquid fertilizer Alpine, but this time not in different rates, but I used the fixed rate, that is the 35 kg of nitrogen per hectare. And so I used the two microbes which I spoke about, the urease inhibitor, and then I used the nitrification inhibitor as well. And in terms of the field gas collection or assessment, we looked into carbon dioxide, nitrous oxide, and then methane. And in terms of our treatments, I had a control where no application was made. And in the second treatment, it's only fertilizer. The third treatment, the liquid fertilizer plus the urease inhibitor. I tried to look at different combinations of this. And then finally, we threw in the granular fertilizer as more or less the conventional practice, where we use the conventional granular fertilizer. And so this was an example of a sampling where we go to collect gases from the field. So we have this a pen of tubes with our syringe where we draw some samples of these gas. We keep these chambers on board for like 30 minutes. And then we draw samples at different time points. So in here, we collected our gases for day 0, day 1 to up to day 12. And then we did 3 sampling, and that is time point 0, 15, and then 30 minutes. So after we draw our samples, we send the samples to the lab to run some analysis. And so we use the GCMS. And then now after we've gotten our concentration values, we end up doing some calculation to get the flux values. And out of these flux values, now we try to do some analysis and then return out with our results. So moving then back to the very first experiment that I spoke about using the nitrogen fixing bacteria, where we're trying to see if we could actually draw some lost nitrogen that is already in the atmosphere into the plant. We realized that after we did a trial, we didn't really see that effect clearly, in the sense that we were expecting that once we added the Envita products, we would have seen, I would say, more of the nitrogen being contained in the leaves, but unfortunately, we didn't see that effect. And so from both locations, we saw very similar trends. However, when it came to the yield component aspects, there was something very interesting, and that is it appears as though combining these Envita products with a reduced nitrogen fertilizer enhanced yield, because that was one of the goals, trying to see how best we can reduce the amount of fertilizers we applied on the field. And so getting some results, it became quite interesting. And so it became some aspects that we hope to actually probe a bit more further, because that is one target we hope to achieve, reduce the amount of fertilizers by incorporating some of these enzymes. And we know the end goal is actually yield. And so once we are getting the yield at a very lower rate of fertilizer application, then that seems to be one of the best options. Moving then into the second aspects, that is the greenhouse gas collections. So after our analysis, I actually drew some curves from the concentration values that we had, and then I ran some analysis trying to find some statistical differences. Unfortunately, there were no statistical differences. Despite the very low levels with respect to the nitrous oxides, methane, they were quite low. However, there were no significant differences. However, you could still see some trends, some interesting trends, which translated into the flux values, which I would come shortly and speak about. So generally, it appears as though we were actually observing some high emissions in other field crops when we compare it to what we actually observing on the wild blueberry fields. I went further to explore, like I would say, some of the results from other works that have been published on high bush blueberry, on some other forest species. And you realize that the concentration values were quite high. For example, in the high bush blueberry, when we compared that to what we actually obtaining in the wild bush blueberries. However, there are so many reasons that actually accounts for some of these differences. It could be the amount of fertilizers that were applied. It could actually be so many environmental factors, like I'll say moisture, could be temperature. And for instance, today I was running some correlation values, and there were very significant correlation values when it comes to the flux measurements or the flux levels with the environmental parameters that were actually reported. And so in trying to draw some line on whatever I have established so far, I tried to do some comparison between just applying Alpine as a comparison to the granular fertilizers. And we could see some substantial reductions when you compare just applying the liquid fertilizer to the granular, that is the conventional practice. You could see some level of reductions. Looking at these raw values, you might actually perceive them to be quite low, but when you actually draw them into percentages, you realize that these are quite substantial amounts. And comparing it also to the control, you could see very low levels. However, you could see some fluctuations when we are looking at the CO2, because we still had, I'll say, plant issues in our chambers when we were doing some of these measurements. And so it tends to cause some level of increase or decreases. However, when you look at the combinations with these microbes that we observed, generally you could see very low levels compared to both the alpine, the granular, and the granular that we have actually measured so far. So this is more or less, I'll say, this cumulative estimates of what I actually made of the 6-day measurements that are obtained for my trials. So in trying to bring us to this point, I will just state that plant and soil microbes have a role to play in reducing greenhouse gas emissions. I believe that much more, I'll say, trials and experiments needs to be conducted to more or less confirm, or probably I'll say establish some of these points. And low greenhouse gas emissions were observed using liquid alpine fertilizers compared to the conventional granular fertilizers. And we could see it from the highlighted rows in the table. It was also observed that some gas reductions were achieved using ureas and eutrophication inhibitors. However, these reductions were inconsistent. Inconsistent in the sense that I made mention of 2 different trial sites, but I only represented 1 trial site. And so doing that holistic comparison, we realized that there are some levels of inconsistencies. And as I mentioned, temperature and moisture plays critical role in greenhouse gas emissions in the wild blueberry fields. And today, from my correlation analysis that I made, there were very, very few correlation values as high as 0.8, which is very, very high. So to end with, I will say that to reduce greenhouse gas emission in the future, I think there might be a need to reassess the type of fertilizers and the nutrient management practices that we have adopted in the wild blueberry field. So I would end here and humbly invite the next speaker to carry on. Hey.
Hola. We're not able to hear you.
Okay, what of now?
Yes, we can hear you
All
now.
right. Oh, thank you so much. So let me just circle back then. I will start all over again. Coming last in this stream of presentation, I'll be talking about remote sensing application in wild blueberry production system. And I'll say over the years, the WRP has been conducting a lot of experiments with respect to the use of prescription map to perform targeted application of agrochemicals on wild blueberry fields, which help in reducing the amount of fertilizer or agrochemical that is being applied. And at the same time, we are comparing 2 systems, and then we are looking at the efficiency, the energy use, and as well as the greenhouse gas emission that is associated with a tractor-based spraying system and an unmanned area vehicle system, popularly called drone. And the thinking is that traditional blanket application of agrochemical is not sustainable, it's waste chemical, and it is not environmentally friendly. And we are also looking at how we can improve on optimizing yield, protecting the environment by using remote sensing to perform targeted or spot application of agrochemicals on our wild blueberry system. This shows a typical wild blueberry field, and you can see we can have blueberry canopy in red here. We can have patches of bare surfaces, and we have weed, patches of weed also on the field. And the thinking is that we only want to apply nutrient management to blueberry canopy while leaving out the bare surfaces and leaving out the weed without applying nutrient management. And with respect to the use of remote sensing, we could use the spectral characteristics of the image produced by a mapping system to actually discriminate between the blueberry canopy and the other kinds of features that we can find on the white blueberry field. And we can also use the structural characteristics, such as the three-dimensional characteristics of the different features on the field, to actually map out the location and the dimension of these different kinds of features that is well seen on the wild blueberry field. For example, we are able to use the three-dimensional structure of goldenrod wheat to discriminate the location of where it is growing on our map, and we are able to map it successfully with high level of accuracy using a LiDAR sensor. And apart from that, we can also use the textural characteristics of all the different kinds of features that we have on the field to also map their location and dimension. And then I must say that timing is actually essential to be able to successfully collect high quality imagery and be able to discriminate between the different features that we have on the field. So we have a kind of setup because we want to compare the performance of a tractor-based spraying system and a drone sprayer. And then we have two field setups at Kemptown and L-Town with 6 different kinds of strips. The nutrient management is Alpine is applied, and it was applied twice in June last year. And this is the system for mapping the field. We have DJI Matrix 300, a very strong UAV with respect to being able to carry multiple payload. It has a maximum flight time of 55 minutes, allowing us to map large area in units time. And we have a combination of different camera and sensors. We have the Zemos P1 and Mica Sense Red Edge, which allow us to capture imagery not only in the red, green, and the green band, but also in the red edge and the near infrared band, which allow us to actually study the plant characteristics. And then the sprayer system, we have the tractor-mounted sprayer system, which actually have a boom width of 6.4 meters and total nozzles of 12 meters. It has Raven Viper 4 control system, and it helps us to apply the prescription map as it helps us spray the agrochemical with respect to the prescription map that is actually loaded on board. And the sprayer drone is DJI Agras T40. It has a payload of 40 kilograms for housing the Alpine that we wanted to spray. The maximum takeoff weight is actually about 101 kilograms. The flight time spraying is 6 minutes, and then we can actually recharge it in 9 minutes. And then if you have a copy of battery, a continuous operation is actually ensured. We also have DJI RTK2 that ensures centimeter level accuracy, so that where we access to spray is actually where the agrochemical is actually deposited. So areas of this are conducted with the mapping drone system, and then overlapping area photographs were collected, and these were processed into digital automosaic. And after that, it was further processed into what we know as normalized difference vegetation index. And furthermore, further processing can actually now help us to classify into blueberry canopy, weed section, and bare surfaces as we found on our blueberry field. And we can actually use different kinds of classifier at this point with respect to the quality of the image, the resolution, and some other characteristics that is inherent in it. The layer is now further classified into a spray zone and no spray zone. The blueberry canopy actually form the spraying zone, and then weed and bare surfaces constitute the no spray zone. Then the layer is graded into 2 by 2 meter, and then it was prepared into the respective format that each system supports. This is a snippet of how the prescription map looks like. And from the prescription map, we are able to insert the application rate. Then we can also estimate the amount or the volume of fertilizer that needs to be applied into each of the trial plots. And then the UAV were actually used to spray the drone, to spray the strip plot. And likewise, the tractor-based spraying system were also used to spray, because we actually want to compare both systems. Other things that we measure is that we try to assess the spray distribution and penetration using water sensitive paper. We calculated the time, the energy, and then the emission associated with the system. Furthermore, we decided to compare the results with a high efficiency spraying system such as the John Deere R4023 sprayer, self-propelled sprayer. It has a boom width of 24.4 meters. It is actually high efficiency system as compared with the other tractor. We are able to have this first set of results, which indicated that the drone or the UAV completes the spraying system in far less time as compared with the tractor-based system. And we also noticed we are able to see that the prescription map, the amount of fertilizer deposited by the drone is actually more aligned with the prescription map, with what is specified in the prescription map, and it's more there less deviation with respect to the amount of fertilizer that it's deposited. We can also see that the prescription base actually allow us to actually monitor closely the amount of fertilizer that we are actually putting up on each of the strip. With respect to the distribution and penetration of the agrochemical that is being sprayed, we realize that the UAV still perform better because there is a better distribution of the agrochemical at the top of the canopy, at the middle of the canopy, and at the bottom of the canopy, in comparison with what the tractor-based system provide, probably due to the advanced nozzle system that is available to us on the UAV. With respect to the emission, we actually collected a lot of data, but I'll only be highlighting the result as with respect to the nitrous oxide. The UAV released far less nitrous oxide in comparison with the tractor, possibly due to the fact that the sources of energy used is actually different, and the system actually performed very much better than the tractor. And with comparison with the high efficiency tractor, we are able to see that the UAV consumes 50% less energy than the, there's a mistake here, I'll just go over it. The UAV consumes actually less energy than the tractor, but it actually also releases far lower amount of nitrous oxide with respect to, in comparison with the tractor system. And it is actually due to the fact that the UAV uses a rechargeable battery, and it was recharged on the field using a gasoline power generator, whereas the tractor system actually uses diesel as fuel, and it actually brings in a sharp difference between the amount of emission and the amount of energy that these two systems use. And as a summary, we realized that the sprayer UAV actually completes the spraying operation faster than the tractor. It uses less energy. It is more precise, and it is more precise with respect to the amount of agrochemical that is required to be deposited on the field. And also the dispersion and the distribution of the agrochemical is actually more intense at the top, mid, and bottom level of the plant system when it is being sprayed. But the use of the drone actually still has its own challenges today. It is still more susceptible to drifting because the flying altitude is a little bit higher than the tractor-based system. We are flying the drone at a minimum permissible altitude of 1.5 meters, whereas the tractor is spraying at a lower altitude of around 0.85 meters. And then the prescription map that the UAV obeys is a little bit coarse with respect to the spatial resolution. A region of no spray zone has to be up to 9 meters, but unfortunately, the features that we map on the blueberry fields are in the centimeter range. So this is another drawback, because some of the features that constitute a no spray zone, the drone cannot really identify it as a no spray zone because their dimension is just too small. Then furthermore, the bottlenecks that is still associated with the use of drone is some of the requirements that is needed to be able to fly a drone in Canada. As of today, if you want to fly DJI Agras T40 in Canada, you are required to have advanced pilot license, as well as what we call special flight operation certificate before you can fly a drone that has a maximum takeoff weight of 110 kilograms. And lastly, it is still not permitted for someone to spray pest control products in Canada. PMRA says read the label, and if you read the label, you need to see RPA, which stands for remotely piloted aircraft, or UAV, specified on the label before you can actually, spray such agrochemicals with a drone like DJI Agras T40. And we take a look at the common agrochemicals that we used on blueberry fruit today, and I think there is really none that has certified the use of drone to actually spray the chemical. We are looking up to the review of the regulation. We are looking up to a relaxation on the rules, which we actually a completion of some researches that will allow the application of agrochemicals on the blueberry fruit using a drone. But as of today, it is still prohibited to use a drone to spray majority of the pest control products in Canada. And that wraps it up. Then I will hand over to Dr. Percival.
Okay, so thank you, Ola.
All right, everyone's done now, or?
Everyone's finished.
Okay,
Oh,
perfect. All right, we did have a couple questions. I think that you guys did a great job of answering them in the comments. Let me just see if there was one. There was one. From Tobias. I don't know if you had replied to him personally, Dave, but he just said that you had spoke last spring about using sulfur lime Bordeaux mixture to control botrytis in wild blueberries. Is there any efficacy in using the sulfur applications to reduce fungal disease pressure from the soil?
and so I responded back to it, Chloe, and then to the broader group. Yes, it helps reduce the primary inoculum source, especially on the fungal side in the crop year. And so we had one PhD student of ours look at this, and it worked quite well on the monolinea side as well as botrytis. So, and it's also a fairly inexpensive treatment application as well. So it's just a good way just to clean things up first thing in the spring.
Perfect. And then I think all the other questions were answered kind of by all of you guys in the chat. So if anybody else has any questions, then feel free to unmute yourself and ask away.
I've got a question for Ola. In your presentation, you mentioned the UAVs gave a more accurate application of agrochemicals. I'm just curious how exactly you are measuring that. And the only reason I'm asking is I was to a presentation in Ontario back in December, and they actually claimed the exact opposite. They're claiming that UAVs behave more like a plane, that there is no downwash and that there's more off-target application because of the low level. So I'm just trying to get a better handle on these and how they work.
Okay. Can you hear me?
Yes.
Yeah. The drone we use actually, we are flying very much lower. We are flying at just 1.5 meters above the blueberry canopy. And you agree with me that distance is actually very closer to the blueberry canopy in comparison with aircraft if you are spraying with an aircraft system. And there is actually a downwash. The propeller is actually powerful, and it is actually helping to push down the agrochemical as it is being sprayed. If you take a look at the video in the slide, you can see that it's actually pushing everything down, and that helps also the penetration of the agrochemical into the plant structure, as the case may be. And this is how we measure how accurate it is. We are spraying at 187 liters per hectare, and we know the area we are spraying. With that, we can calculate the volume. And when we estimated the amount of fertilizer that was deposited in the area, it is closer to the expected amount at the end of the day. Unlike the tractor-based system that we have a kind of over-application in some places and some under-application in some places, and the deposition of the agrochemical is not as regulated as what we have with the UAV. So we have a closer control with respect to the amount of the application rate and the amount of agrochemical with the UAV than the tractor-based system in our experiment.
Okay.